Hello guys, welcome back to another exciting lecture of Inside Learning. So today we are going to discuss about the structure of DNA. It is the most widely accepted structure of DNA and how it formed and how it was considered the most acceptable and the most refined structure we will see in this lecture. So let's just begin our lecture. We are going to discuss about Watson and Crick model of the DNA. So what happens in 1953? Basically in 1953 there were two scientists that are named as James Watson and Francis Crick. They just combined the physical and the chemical properties of the DNA. All the data that they have at that time, they just combined their physical and chemical nature, and they proposed the double helical structure of DNA. So at that time, that was the most acceptable, and till today we are widely discussed and study this structure of DNA. So they just proposed the double helix structure of DNA with these properties in 1953. So what is the double helix nature of the DNA? Basically, the double helix nature of DNA is that the two polynucleotide chains that are twisted in spirals. So what is polynucleotide? Poly means many, nucleotides mean made up of proteins and DNA. So there are two chains made up of polynucleotides that are twisted together, that are twisted in spiral like this way. As we spiral or we twist the two threads like in this way. This is the structure of DNA. Basically here we are talking about these two chains. Therefore the DNA molecule is in the form of double helix. Stacking of the nitrogenous bases. So what are the nitrogenous bases? Till we see that polynucleotide chains, what are those chains? These spiral chains or the chains that are collide with each other. And we also have a structure known as nitrogenous bases. Nitrogenous bases are basically the specific type of bases that are present or enclosed inside the spiral structure of DNA. The sugar phosphate molecule form backbone of the polynucleotide chain. So basically the two chains, the spiral chains that are polynucleotides, they are made up of sugar phosphate. Sugar means there is a presence of carbohydrates and phosphate means there is a presence of phosphate group. The base pairs, here we are talking about the nitrogenous bases, extend form of the sugar phosphate backbone and are stacked inside the double helix. As I have already told you that the nitrogenous bases are just stacked or trapped inside those two chains. I am going to show you the structure of how exactly it looks like. This is the double helical structure. This is one chain of the DNA strand, or you can say DNA strand, and this is another DNA strand. And here you can see that nitrogenous bases are present. The double helix executes a two turn every 10 base pairs. So basically what happens that after every 10 base pairs, like 1 base pair, 2 base pair, 3 base pair, this is exactly like after every 10 base pair there is a turn in the double helix. So this is a very interesting fact that after how many base pairs there is a turn or there is a twist on the structure of the DNA strand. So after every 10 base pair there is a turn and the double helix structure just twisted around each other. So base pair, now we will talk about the base pair. Here first I want you to see that how many colors are there of base pairs. I just intentionally painted them in such a way that you can easily distinguish between them. Like we have orange, pink, yellow and green color in base pairs. You cannot see any other color other than these four. Okay, so these four base pairs are basically representing each of the nitrogenous bases. There are only four types of nitrogenous bases present in the eukaryotic human cells. First one is the adenine form, which form the base pairs with thymine. So there is a presence of nitrogenous bases adenine, thymine, guanine and cytosine. These four are the nitrogenous bases. And how they form bond with each other? Adenine always form a bond with thymine and guanine always form a bond with cytosine. It was observed that these bonds are known as hydrogen bonds. Base pairs are combined together, they just stick together, and adenine always form a bond with thymine and guanine always form a bond with cytosine. And which bond they form? They form the hydrogen bond. Like in case of proteins form the polypeptide bond. So let's just watch it very carefully. Here you can see three lines. If you can watch very carefully, here are the three lines. Here you can see two lines. So the hydrogen bonds that are present between guanine and cytosine, they are three in number. Yellow is representing the guanine and pink is representing the cytosine. Guanine and cytosine form three hydrogen bond, while adenine and thymine form two hydrogen bond wherever they present. They form triple bond and they form double hydrogen bond. So these are some very important MCQs, like how many bonds, hydrogen bonds are formed between cytosine and guanine, and after how many base pairs there is a twist on the DNA structure. So just keep these things in your mind. So now just discuss about little bit more about the structure of the DNA. There are two different grooves. So grooves are basically this part of the DNA, this part. This whenever the DNA double helix structure twisted, the structure or the appearance they form, it looks like a groove. So there are two different grooves. The larger grooves is the major groove, and the smallest one is called the minor groove. So what are their functions? Basically they express the genetic material. Obviously we are discussing about the structure of DNA, and DNA is all about genetic information. So these grooves are always and also expressing the genetic information. Involves in interactions between the double helix. It is always obvious that when the twist form around the double helix structure, it interacts, or it helps the double helix structure to stay in contact, to stay the two chains in contact with each other. The anti-parallel arrangement. So what is anti-parallel arrangement? The strands run in opposite direction. If orientation of one chain is 3 to 5, then the other will be 5 to 3. So it is very obvious that the strands are in opposite direction. Like I would love to show you here that suppose it is one strand of DNA, these strands here, they are twisted around each other. Now I am going to show you in a ladder form. This is one strand of the DNA, and this is the other strand of the DNA. Right now I just open them so that you can easily understand them. They are run in opposite direction. What is mean by that? If the one strand is in 3 to 5 direction, okay, so there will be a presence of A, G, let's suppose C and T, okay. If the other strand of the DNA is run exactly in the same direction, for example if it runs in same direction like 3 prime to 5 prime, it will be A, G, C, T, and it never possible. A always form a hydrogen bond with T, not A. G always form a hydrogen bond with C, not G. So they have to be in opposite direction so that they are complementary to each other. Now if 5 prime and 3 prime opposite direction of the strand, then there will be T. For G there will be C, for C there will be G, and for T there will be A. So in this way it will run exactly the right way. So they are anti-parallel arrangement of double helix structure. They are opposite to each other. Now what are the complementary of the sequence of one determining the sequence of the other. Like I have already told you, and I have already shown you that sequence of one strand determines the sequence of another strand in this way. A and C of one strand correspond to T and G of the other strand. A and C of one strand correspond to the T and G of another strand. Obviously the one strand determines the how would be the sequence of the next strand, so that they form the hydrogen bonds and the double helix structure of the DNA ultimately. So this is how the structure of the DNA forms. They are complementary to each other. They are anti-parallel. They have the two major grooves, and they have a very basic and important function. And they have the base pairs, how these base pairs are combined together, and what are their numbers. Also, in which year they just discovered this structure. And also don't forget about the sugar phosphate backbone, which is very important. So there are numerous MCQs related to this topic. Next we will discuss about some very important short questions about this topic, Watson and Crick model. So this is all about today's video. If you like my video, do subscribe to my channel and share this video with your other fellows also. Bye bye.